help me welcome Dr. Pesek.
Thank you, Steve. Well, it's a pleasure to be back in Wilmington. It's actually our third visit here. And so we're getting to know the area, and hopefully we'll know it a little bit more in the future. So today I want to tell you a little bit. It's kind of a long story. Actually, if we've made it in years, it would be 25 or more, on the development of what we refer to as aqueous normal phase chromatography. And it's based on a particular type of material that we developed in our laboratory over this period of time. So what I want to do is describe how aqueous normal phase chromatography works and show you why it's different than if a technique that's also used to separate polar compounds referred to as hydrophilic interaction liquid chromatography, and kind of give you a hint of why this particular method has a lot of advantages to it, and then show you some kinds of applications, right? It's great to look at something from a theoretical point of view, which I'll try to describe here as well, but it's also good to prove to you why it's useful. Hope I'll accomplish that. So here we go, a little bit about aqueous normal phase chromatography. It has what's referred to as both reverse phase and normal phase retention. So I'm going to define that here. This is, on this left-hand axis, tells you about how well a particular compound is retained. And there's a mathematical term for that called a retention factor. And on the bottom is the composition of the mobile phase. And this is shown in percent organic of the mobile phase. So if hopefully most of you know a little bit about chromatography, reverse phase generally is used to separate nonpolar molecules, and the mobile phase has to be somewhat different than the stationary phase. So we have to have a large amount of water in the mobile phase. So over on the left-hand side, where we have a lot of water, we have hydrophobic compounds being retained. And normal phase, generally used for more polar compounds where you have a higher amount of organic. So there are a number of different ways in which compounds can be retained, and you can look at how these materials work. One, of course, is to have a case where you have compounds which are either on one side or the other, right? You can only retain them by the reverse phase mechanism. So it means you've got a lot of water. On the right-hand side, you have molecules that can only be retained in the normal phase mechanism. So you have a lot of organic, and these are the hydrophilic molecules. Then you have some cases where there's overlap. You may have some compounds that can have appreciable retention under intermediate amounts of water and organic, where both hydrophobic and hydrophilic compounds can be retained. There's an and this can move a little bit to the left or right, up or down. It all depends on the types of compounds that you have in your mixture. And then you have some compounds, generally larger molecules, that have both hydrophobic and hydrophilic components in them, right? They have a polar group on it, but they have constituents that are hydrophobic as well. And so those could theoretically be retained by either mechanism. The reverse phase, if you're emphasizing the hydrophobic aspects of the molecule. Hydrophilic, if you're looking at the functional groups, which can be polar. So the beauty of the material that we developed over this year is that it works in both mechanisms. So that's hopefully I'm going to try to show you here. And what is the material that we work with? Well, most chromatographic stationary phases are based on ordinary silica, which the surface, this is a polymer of silicic acid. So at the end, the terminal groups are OH, silanols. This is very polar. It tracks water. In fact, you know, if you get a typical electronic device, you have those little packets in there to keep the components dry. That's filled with silica to adsorb water. The material that we've developed is referred to as silica hydride, and the base structure is very similar. In other words, it's got siloxane linkages all over. That gives it the mechanical properties that are similar. The two materials are similar from a mechanical point of view, but the surface groups are SiH, which is actually mildly hydrophobic. So just from a chemical perspective, you'd say, well, these two don't operate under the same conditions, and that's true. Here, with the very strong polar functional groups on the surface, you generally get strong adsorption of very polar compounds. And the innovation or innovative development in chromatography was to modify these groups here to make them less hydrophilic, less hydrophilic, less polar in nature, so that you could retain polar compounds. However, even under the best of circumstances, no matter how good of a chemist you are, you can't modify all of these groups. There's a lot of steric geometrical considerations that make that impossible. So there's always going to be a large number of OH groups on the surface. The good thing about this material is you can modify it as well, but when you're finished, you still have SiH groups on the surface rather than hydroxide, so these are mildly hydrophobic. Okay, so this is the way that you modify the silica hydride. It's a reaction called hydrosylation. It's been known for a long time. And it's a fairly simple mechanism. You react the SiH group with some group that has a double bond, usually a terminal double bond or a terminal triple bond, with the catalyst, and you get a direct silicon-carbon linkage, which is a very strong bond. That's another advantage. The catalyst, well, there's two types of catalysts. You can use a metal complex like hexachloroplatinic acid. That's referred to as Spear's catalyst. But there are other metal complexes that work as well. Or you can do it by free radical initiation with things like T-butyl peroxide. So you have two mechanisms for being able to attach molecules to the surface. With silanols, almost everything is done by a reaction referred to as organosilanization. So you have only one way of modifying the surface. Okay, so this is some work from a laboratory in the Czech Republic using the material to sort of verify the dual retention capabilities of the silica hydride material. These are just two different stationary phases. One is C18, which is, again, the most common material used for reverse phase separations. It's just a straight hydrocarbon chain, 18 carbons, and a material we call the diamond hydride, which is basically silica hydride with a small amount of carbon on the surface. But it shows you here that both of these materials, whether we have a big C18 group or we have a very small amount of carbon, operate in both mechanisms. So it doesn't make any difference what you put on the surface. You still retain properties for retaining both hydrophobic and hydrophilic compounds on the surface. Okay, so here just shows an example of two different runs, one done in the reverse phase mode, one done in the normal phase mode. So figure A, give you a little bit of chromatography nomenclature here. The solvent A always refers to water. Solvent B refers to the organic constituent. And in this case, solvent B is acetonitrile. So for figure A, we have a very small amount of water and a very large amount of acetonitrile, 85%. So we're operating in the normal phase mode. And you can see that we're able to separate these four compounds. In figure B, we start out with a small amount of acetonitrile. So that means we're operating in the reverse phase mode. And you can see that, again, we don't have exactly perfect separation. These weren't completely optimized. But you see you have different retention orders. You have different degrees of retention between the compounds. So, but you can tell that this material is operating in both modes. In this case, normal phase mode, in this case, reverse phase mode. So again, verifying the fact that whatever silica hydride-based material you choose, it operates in both modes. Okay, here is just pure silica hydride itself, verifying the fact that the surface is mildly hydrophobic. So here's uracil, which is a very polar compound. It's not retained at all, so the retention factor is 0, meaning it's not retained at all. Pyridine has some hydrophobicity to it. It has a small retention factor. Phenol is a bit more hydrophobic, so stronger retention factor. A couple of more over here, acetophenone and biphenyl, also having some retention on a pure silica hydride surface. So this verifies the fact that if we look at it from a chemical point of view, the surface itself is mildly hydrophobic as compared to if you did this on ordinary silica. All of these molecules would have no retention at all. Okay, so this is the C18, and the most common type of separation used, reverse phase. And so if we bond a C18 to the surface, we can do that as easily as organosilonization. We get very good retention of all these different types of hydrophobic compounds. And these are gradient separations. And you can see that this bottom statement here showing that these are 2 different column lots. So this is to verify that we can make the material reproducibly. So you have to look very hard to be able to tell that there's actually 2 separate chromatograms there. So they overlay each other very nicely. So we're able to accomplish what you can with most stationary phases. That's always good to do. You want to be able to do what everybody else can do. What else can you do? Well, here's C18 again. Here's a very, very polar molecule, methotrexate. It's used in cancer chemotherapy. It has amine groups on it. It has carboxylic acids. And we're going to try to retain this on a surface that's been modified with a very large hydrophobic molecule, C18. So we're going to use, again, the, what we call the inverse gradient, because we're working in the normal phase mode, a large amount of solvent B, which is the acetonitrile, solvent A, DI water, plus a small amount of formic acid. So when we do this, you can see that the time of a compound not being retained would be down here about 1 minute. So this is over 9 minutes. So this very polar molecule, even though the surface theoretically is modified by a very strong hydrophobic compound, C18, still retains in the normal phase mode. And I can guarantee you that if you can take any other C18 stationary phase made by organosilonization, it will not retain this molecule. All right, so now we have to figure out what's the difference between ordinary silica and silica hydride. Something fundamentally is different about it, so to be able to explain this property. Again, this was some work that was done by another laboratory. And what they did was they measured the layer of water on the surface. So here's our 2 little cartoons that show us a little bit about this. This is ordinary silica, which we refer to as a HPLC material, a hydrophilic interaction liquid chromatography material. And it has a very, very thick layer of water on the surface, even though it's modified by C18 groups or other groups. On the other hand, the silica hydride, again, if you look at this column here, this is the important piece of information on this slide, shows you what the average thickness of the water layer on the surface is. And you can see that in the top 4 materials, these are all things that are based on ordinary silica. And they have anywhere from 3 to almost 10 layers of water on the surface. If you look at silica hydride, these are the 4 materials on the bottom. This is our cartoon. They have less than a half a monolayer of water on the surface. So even though we have the same group onto the surface, we have these small patches of water that are on the surface. Here we have a very thick layer. So this is the first fundamental difference that we were able to find between the two materials. That tells us something about it, but it doesn't explain the whole story. So what does the water layer mean, chromatographically? Some things we can already say in comparison to these ordinary silica materials and silica hydride. One thing that we noticed, even before we did the water experiment, was that if we change the mobile phase composition, the silica hydride equilibrates very, very rapidly. You can do another chromatographic run in just a very short period of time. But if you have an ordinary silica material, it can take many, many minutes, or sometimes people refer to these as column volumes, how much liquid we got to pump through the column before the thing equilibrates, and you can do another chromatographic run. So this is one noticeable difference between the two, and we think this is because of the water layer. If you have not very much water on the surface and you change the mobile phase composition, it doesn't take very long for those few molecules or partial monolayer to re-equilibrate on the surface. But if you have 3, 6, 9 layers of water to reform on the surface, there's a finite amount of time for that to happen. And it can be very appreciable in many cases. And people have noticed this for a long time. They've been talking about it and trying to understand, why the hilic material, the ordinary silica, takes so long to equilibrate. All right, so we have to then begin to think, well, there has to be then a fundamental difference between how silica hydride retains compounds versus how ordinary silica retains compounds. And after what people have been working with the ordinary silica materials, referred to as the hilic, for a while, and they've come to the conclusion that the mechanism of retention for these materials is a partition between the mobile phase in general and this very thick water layer on the surface. It's more polar, right, than a mixture of, say, acetonitrile and water. Pure water is more polar. So the compounds, the polar compounds, are attracted to this thick layer of water, and they partition between the two. And that's it's been sort of the generally accepted mechanism for that. And this slide here just shows us that this water layer, as it becomes thicker, and if we increase, for example, if we put a buffer in the mobile phase, if we increase its concentration, the components from the buffer also preferentially go into the adsorbed water layer, making it even more polar. And most compounds that are hydrophilic begin to retain longer. That's a very well accepted observation about what happens with the silica materials. But on the other hand, for those compounds, the silica hydride, which operate in the aqueous normal phase mode, we see the opposite effect. We begin to see that as we add more buffer to the system, instead of the retention increasing, it decreases. So it's another hint that we have some fundamental difference between the two materials. We've already shown that the water layer is different. Makes sense that as we may increase some ionic component in the mobile phase, it would preferentially go into the adsorbed water layer. And of course, when we're dealing with compounds that are going to be retained in this water layer, if we make them more polar, then we're going to get increased retention, the opposite effect. You may think that may not be that important, but it actually is from a practical point of view, because most people, or an increasing number of people who are using LC are using LC in conjunction with mass spectrometry as the means of detection. It's much more specific, in many cases more sensitive than other types of detection systems. However, if any of you have ever run an LC before, one thing you notice is if you're running a solution through your ionizing medium for the mass spectrometer, if you have a lot of salt around, it begins to condense. on your ionization source. And you got to clean it very often. But in the case of aqueous normal phase chromatography, we can use less salt and get more retention. So the mass spec source is clean for a much longer period of time and is much more reproducible. So it has a practical effect too. So summary at this point is that if we're talking about retention in the reverse phase mode, it's very similar in most cases to other stationary phases that are available commercially. There might be some selectivity differences due to the fact that the hydride is on the surface, but basically this operates on the basis of hydrophobic interactions. So there's minor differences, but not major. The interesting thing is there's definitely a different mechanism operating for the retention of polar molecules, and the fact that the surface seems to be mildly hydrophobic. How do polar molecules get retained on something that's mildly hydrophobic? That was the big question to answer. We could say, okay, we can prove that there are differences between the two, but how do we rationally explain the fact that we can get retention of polar compounds on a hydrophobic surface? So first thing we did was we measured with our colleagues in Australia what's referred to as the zeta potential. And the zeta potential, from a simplistic point of view, is just what's the overall charge on the surface. And as a function of the amount of acetonitrile in the liquid surrounding it. And you can see that as we increase the amount of the organic component, the charge on the surface gets more negative. You can look at the scale here. It gets more and more negative as we increase the amount of acetonitrile. That's very interesting. From a hydrophobic surface, why does the charge become more negative? So that was the question to answer. So what's responsible for that? Can't be silanols. We've studied this material a long time ago very extensively. And by NMR techniques, you can do this by silicon-29 or proton NMR techniques, you can prove that there's no more than 5% SiOH groups left on the surface of silica hydride. So ionization can't be due to silanol groups. So we started searching around in the literature trying to figure out an explanation to this, and we came up with a very interesting series of papers that we found. Some people did some experiments on oil drops, and they put oil drops into a solution, a water organic solution, and they discovered the same thing. The surface of that oil drop became negatively charged. So we said, okay, well, we have to, maybe this is a good model for us because remember, silica hydride is basically hydrophobic, so it kind of acts like an oil drop maybe in an aqueous solution. And so we began to pursue this a little bit further to figure out a good explanation for it. And this negative charge is due to hydroxide ions, which come from the autodissociation of water. Now you may say, well, there's a lot of organic solvent around, but there's still a lot of water. Even at 10% water, that's still a lot of water in comparison to the surface itself. And so the autodissociation of water has a potential for being able to have negative ions on the surface. The positive ions bury themselves inside, the negative ions are on the surface, and that's basically responsible for the negative charge on the surface, and it's why hydrophilic compounds are retained, because there's this negative charge on the surface. So here, and if you forget this one, there's a graph over here that allows you to look at it again. This is our model, right? We got the silica bead. It's got the hydride surface on it. So it's got this negative charge. And this is the overall region of the measurement of the zeta potential. So it definitely encompasses the whole surface of the silica bead and accounts for the overall negative charge that's there. So when we have compounds. Here's some polar compounds. These are analogous, chemically analogous to AMP, ADP, and ATP, these are very polar because they have phosphoric acid groups and the nitrogens on them. So when they approach the surface, they're attracted to this negative charge or displace some of the negative charge on the surface, interact with it, and that's how you get separation. So this is the fundamental difference from, again, ordinary silica, because if you go ahead and look at the negative charge on an ordinary silica surface, it's due to the silanol groups. But in the case of silica hydride, it's due to these hydroxide groups which have attached themselves to the surface. So that means that if you have a negatively charged surface, if you have a positively charged compounds like the ones we have here, cyanoguanidine, melamine, and metformin, you have charge attraction, right? If you're, in this case, the mobile phase has in it some type of acid like formic acid or acetic acid, these nitrogens are going to be charged and they're going to be attracted to the negative charge on the surface. And so that's responsible for these separation of the hydrophilic positively charged molecules. You see, we get very nice separation between these three compounds here. Melamine you may know about from the cases where there's been pet food and baby food that have been spiked with melamine to increase the nitrogen concentration to make it look like it has more protein in it. Metformin actually is a common drug. And cyanoguanidine is actually a decomposition product of melamine. So we get nice separation. The bottom one here is another thing that we like to point out about the silica hydride materials. I showed you the fact that lot-to-lot amounts of material are reproducible. This is an example of just doing the same run over and over again. And again, this is 4 overlaid injections. You can see very, well, not very easy to see that there are 4 because they completely overlap each other. So this is, again, one of the examples of why silica hydride is different. And this is a gradient run. So that means the mobile phase is changing with time, going from 100% of the organic constituent down to 20%, and then we go back up. And typically, the amount of time that it takes to, before we do the next injection is a few minutes. If we were doing the same thing on a silica material, it would be 15, 20, 30 minutes. It would not be uncommon for this type of an analysis. But this we generally do in maybe 2 to 3 minutes. Okay, and of course we can retain other types of polar compounds. This is maleic and fumaric acid, which are the same chemical composition. They just cis and trans isomers of each other, and we can separate those very nicely on the silica hydride material. Here's a couple of other compounds that are potentially negatively charged. This is probably done by some type of displacement. Displacing the hydroxides on the surface with the negatively charged components in these particular compounds. So we have two different mechanisms, both sort of like adsorption mechanisms, definitely not partition because we have a very small amount of water on the surface, less than a half a monolayer. And here's another group of compounds. These are phospholipids. This was again done by another laboratory. They were unable to separate this very complex mixture here at the top using the Hillic material, but on the silica hydride material, they got excellent separation of most of the compounds and were able to do some good quantitative work as well. This material is the cogent diamond hydride. This is, I mentioned before, is a silica hydride surface with just a tiny amount of carbon on the surface. It seems to help in producing better peak shapes. So it's a very good material for separating a wide variety of polar compounds. People use it for all kinds of very interesting applications. Here's another one, histamine, which is a very common component in many types of wine. Here's the standard up here. And you see that most of the detection we do is by LCMS. So you'll see along the side here a mass to charge ratio. And when it shows you this many significant figures, it means we're using a high resolution mass analyzer like a time of flight. And so this is the standard. This is white wine. And you can't see any histamine in white wine. It's not very common in most white wines. But in a lot of red wines, it is a very common constituent. And here you can see it very easily in this particular red wine. And by a lot of analyses, we determined that many California red wines are fairly high in histamine. But on the other hand, if you're allergic to histamine, the good news is you can drink Italian red wines because they're low in histamine. So if you have any histamine problems, go out there and buy those Italian wines. What else have we done? Well, here's some sugars. We're doing this on a column that has been modified with an amide group on it, which has been known for a long time to be a useful way of separating sugars, but it's not an easy thing to put on a surface. So here's ribose and xylose, which we can separate very nicely on the amide column. You know, we have also here a couple of other compounds that we've used for separation. This pyrilamine and 4-amino-3-chloropyridine. Also, because of their nitrogen-containing constituents, can be separated quite nicely on this amide column. But if you look on other types of materials, they're not separated. Here's a compound example that we actually are using now in one of our laboratories, in our quantitative analysis laboratory. We developed this one for the components in the uric acid metabolic cycle. And so that's allantoin, 6-amino-uracil, and uric acid, and tyrosine is our internal standard for doing quantitative results. So this is the standards, and this is a real sample for being able to detect those 4 compounds. And you can see the 3 compounds, of course, from the uric acid cycle, and the tyrosine that we use as the internal standard. This is done on a surface modified with the diol group. So it's got some carbon on it, of course. It's an organic molecule, but the functional groups are 2 OHs. This is also done in the normal phase mode because we're starting with 95% B, the organic constituent, which is acetonitrile with 0.1% formic acid, and water, which also contains formic acid. So this you can do as a very practical experiment to run, and like I say, we're using it right now in our quantitative analysis laboratory. Now this is another interesting application that we worked on with our collaborators in Australia. We took a series of peptides and of varying properties, and the distinguishing property that we're looking at here, is the hydrophobicity. And you can calculate how hydrophobic a molecule is. And this number, if it's a very large positive number, then it's a very hydrophobic compound. If it's a negative number, then it's hydrophilic. So we're spanning a broad range of hydrophobic, hydrophilic properties in these peptides. And these are the PIs, so they're considerably different from each other. And what have we done? Well, we've separated those 7 components, not perfectly baseline in this case, but we're more interested in understanding a bit more about the properties of the material. But in a single run, it's a gradient run, in the normal phase mode, we are retaining and pretty much completely, except for this one pair here, 2 and 4, separating a series of peptides that go from extremely hydrophobic to very hydrophilic. It's almost impossible to do this on any other type of stationary phase. Wouldn't be done. So, and you can see that the hydrophobic peptide here, which is the largest positive number, has appreciable retention. There's the void marker right there. So this shows you that you can separate compounds of both hydrophobic and hydrophilic nature in a single run, kind of like we postulated in that first graph that we looked at, which showed that, if you hit the right mobile phase composition, you can retain both hydrophobic and hydrophilic compounds in a single run. So this is a good verification of that. First time that, that I think that we've proved that this is for a really challenging type of separation will work nicely. Okay, and this is another example. So it stimulated us to try some other examples. This is another one of separating two compounds, one which is hydrophobic and one which is hydrophilic in a single run. And this is phenylglycine and ceretylene. One, of course, this compound number 2, because of the of the greater number of nitrogens and also the chlorine groups, make it a little bit more amenable to analysis or retraction to the hydride surface, and so it's retained a little bit longer than the phenylglycine. But again, if you calculate the hydrophobicity of the two, the sertraline has a P of about 2.9. And this is done still with the fact that it has longer retention than phenylglycine. So it's an interesting combination of the two. Okay, and one other material that we've made quite a number of years ago is to bond cholesterol to the surface of the silica hydride. Cholesterol is an interesting compound. Obviously, it has lots of physiological implications to it. But from a strictly material point of view, it is what is referred to as a liquid crystal. A liquid crystal is, in its free state, has properties of both liquid and crystal. In other words, it has liquid properties to it, but it has a preferred orientation in the liquid state. So if you heat up cholesterol so that it becomes more liquid-like, those molecules are not free to tumble and rotate. They have a fixed orientation. We decided, well, maybe it'd be interesting if we bonded this to a silica surface and see how it behaved from a separation point of view. So this is the material, the cholesterol material, and it separates by the basis of molecular shape. Because when you put the cholesterol on the surface, the molecules tend to retain some of their liquid crystal properties, meeting the orientation. And so they tend to line up very nicely on the surface, rather than kind of randomly flopping over here and there. No, they stick straight up like little fence posts. And therefore, when molecules in solution approach it, if you have a long, thin molecule, it goes in between the fence posts. If it's a bent molecule or has a lot of bulk to it, it doesn't penetrate very far. So this is a nice application of that. This is oleic acid, and which is a cis isomer, and the trans component of it, the alidaic acid. So this is straight chain, and therefore it penetrates a little bit further into these fence posts than this compound. So this one gets eluted first, right? Doesn't interact quite as much with the stationary phase, so it comes out first, and the trans component comes out later. And we have a number of other examples of different types of steroids which are based, the separation is based on structural differences in terms of how they can penetrate into the cholesterol stationary phase. Okay, so this, as I mentioned, is a very common problem with helic materials, or non-reproducible retention times. And part of this is due to the fact that the water layer is not easily kept at a constant value, especially between gradient runs. And this is an example of something that someone did in the literature on a helic column. And they can see that as time changed, the water layer was very, very slowly equilibrating, and it took a long, long time, and they still were not getting reproducible retention times. It's a very frequently reported phenomena in Hillock, but not us. This is a gradient run, and this is glucosamine. So this is 10 consecutive injections with approximately a 1-column equilibration time, meaning just a couple of minutes. And again, you can see, like some of the other examples I showed you before, you have to look very closely to see that there's actually more than a single run. It almost looks like only one chromatographic run, but there are 10 peaks superimposed upon each other there. And another example, this is the batch-to-batch reproducibility. This is important. If you want to do, on a routine basis, a certain type of analysis, no column lasts forever. Ours are pretty rugged, but they last. Every once in a while, you have to replace them with a new one. And when you get a new one, you want to make sure it operates exactly like the other one. So here's 3-column batches of the same analysis. This is the diamond hydride, and this is the compound doxylamine. So you can see pretty much all 3 are lying right on top of each other. So this material can be made reproducibly. So some of the differences between now aqueous normal phase and Hillock or ordinary silica material, less than a monolayer of water on silica hydride versus anywhere from roughly 3 to 10 layers of water on Hillock material. So therefore, the composition is not going to vary from run to run or when you have gradients. And this gives you much higher precision in the analysis. Rapid equilibration of the stationary phase is very important. Again, especially if you're doing gradient runs. And if you have complex samples, most times you're going to be doing a gradient run. 2 to 3 column volumes is fairly common. 15, 20, 30 or more sometimes are what's required for an ordinary silica or a Hillock material. The negatively charged surface on silica hydride is due to hydroxide ions. Negatively charged surface on ordinary silica, silanol groups. Totally different. And so therefore we get both differences in selectivity and in most cases enhanced retention in the aqueous normal phase. And of course we have the ability of the silica hydride to function in the reverse phase mode, even if it's not modified. But if you modify it with something that has some hydrophobic property to it, like C18, you get ordinary reverse phase retention. If you use ordinary silica materials that are not modified, you don't get reverse phase retention. We have excellent run-to-run and column reproducibility for the silica hydride materials. Again, it's very important if you're doing routine analysis or if you're trying to repeat experiments that you're doing in a research laboratory. You want to be confident that the data you're getting is going to be reproducible. And the other important part is that you don't have to have high additive concentrations in the mobile phase, which you almost always have to have with Hillock. Very seldom do they operate at less than 25 or 30 millimolar additive. Sometimes 50, 60, even 100 millimolar additive is necessary to get the method to work. And if you want to use mass spectrometry as the means of detection, that's just not acceptable. You're not going to get, you're going to be cleaning your source very frequently. So that means that we're operating at 15 millimolar or less. Typically 5 to 10 is more common, but a few cases we've operated up to 15. And you can work for long periods of time at 15 millimolar and not have to clean your mass spec source. So that's the story. It took 25 years or so to bring you the complete thing. But over the years, we've been very fortunate to get funding from the National Science Foundation and NIH with our collaborators in Australia, the Australian Research Council, and the Keck Foundation, which is a private organization. And of course, we've done some collaborations with Agilent, which are close to us. But of course, our most important collaborator, our friends who are here today, Microsolve Technology Corporation. We've been working with them for 15 years. Went by in a hurry, didn't it? And they're your neighbor. They're in Leland, North